Hey class, in this recording we're going to focus on leukocytes. As we think of leukocytes, I need to emphasize that they are the least abundant. We have hardly any leukocytes relative to all the other formed elements. So it's the least abundant formed element within our bloodstream. But it's one of the single most important. I mean, we get a lot of bang for our buck, I should say, with these leukocytes. They are going to help us help protect us against pathogens, infectious microorganisms that can make it into our bodies. A key characteristic of the leukocytes is that they have a very conspicuous nucleus. I remember when I was younger, a buddy and I were getting an argument about how, in that old movie, that 1993 classic Jurassic Park, how on earth could they isolate dinosaur DNA from the blood that a mosquito sucked out of dinosaurs, because all the red blood cells were anucleate. They didn't have any DNA in the red blood cells because there's no nuclei in the red blood cells. And my buddy pointed out, he was like, hey, what about the white blood cells? Those have nuclei, those have DNA. And we concluded that all the dinosaurs from Jurassic Park must have come from the leukocytes that were sucked up by that mosquito that movie was based off of. Now, back on task here, back on topic. When we think of these leukocytes, they typically only spend a few hours in the bloodstream. They don't spend a lot of time in our bloodstream. They're going to spend most of their life cycle roaming around in other connective tissues of our bodies. Some organelles are going to be retained within our leukocytes because these leukocytes are going to be actively synthesizing proteins. And we can break up the leukocytes into two broad categories, granule sites and agranule sites. All white blood cells, or all leukocytes, will have lysosomes. These are the non-specific granules. The granule sites, though, have very specific storage vesicles in them, or granules, that contain very specific enzymes for specific purposes within our bloodstream, or within our immune system. So as we look at those granule sites, we have the fills, fill. Neutrophil, eosinophil, basophils are the granule sites. Neutrophils are notable in that they are the most common white blood cell. They are also polymorphonuclear. Oh, that's a fun word. Polymorphonuclear. In other words, their nucleus changes shape many times, or there's many shapes to the nucleus of a neutrophil. Generally speaking, I like to think of it as a horseshoe. We have the eosinophils. They are some of the rarest white blood cells. Basophils are the rarest kind of white blood cell. When we think of eosinophils, look for rosy orange granules. Eosinophils are typically going to have many dark violet granules. When we look at those agranule sites, they consist of the lymphocytes and the monocytes. And as we look at that lymphocyte, the lymphocyte is a fairly common, the second most common kind of white blood cell out there. And they are typically going to have not that much, a variable amount, but a small amount of cytoplasm and a very large dark violet nucleus, monomorphic, or one shape, just big round nucleus. It takes up the vast majority of the cell. And then the monocytes are the big guys, the largest white blood cells out there. So let's talk about them in specifics. As we look at the neutrophils, the polymorphonuclear cell, these cells have nuclei that are they're crazy. They have a kitty wampus shape. So if we look at these nuclei, the key thing to focus on is how there's different globs, or different regions of the nucleus, and you can see little tiny connecting branches connecting those different regions. Another thing to focus on with the neutrophils is that their granules, or the speckles, are fairly faintly stained. When we think of neutrophils, I want you to think antibacterial. If somebody has neutrophilia, they have lots of neutrophils in their bloodstream, abnormally high concentrations of neutrophils, and that's a key clinical indicator that that individual is battling some kind of a bacterial infection. As we look at the eosinophils and basophils, we have the eosinophil here on the right. Excuse me, on the left we have the eosinophil, and then on the right over here we have the basophil. Eosinophils are going to focus on fighting off parasitic infections. They are also going to help us with diseases of our collagen and allergic responses and diseases of the spleen and or central nervous system. Eosinophils are specialized in that they can phagocytize antigen-antibody complexes, and they can also release inflammatory chemicals and respond to allergens. They also can release digestive enzymes to the extracellular environment to help neutralize multicellular or large parasites that may be within our bodies. As we look at the basophils are the rarest white blood cell. And notice how the speckles or the granules on the basophil are dark purple, completely obscuring the nucleus. Whereas with the eosinophils, we have those pink granules filling up the cytoplasm. When you think of basophils, I want you to think of a cell that's going to be associated with secreting histamine. And that histamine is going to be acting as a vasodilator so that we have increased rate of blood flow to injured areas. Basophils clinically have been associated as a white blood cell that will increase in numbers when we have a person with a chickenpox infection, sinusitis, so sinus infection, and or diabetes. In addition to secreting histamine, basophils also secrete heparin, which acts as an anticoagulant. So basophils can make it so blood can flow faster and so the blood does not clot. In other words, they make it so we can move blood really efficiently because the blood becomes less viscous and the blood will have a bigger tube to travel through. And we also have the agranule sites, the cells that do not have conspicuous granules within their cytoplasm. Lymphocytes are the most common agranule site. The lymphocytes have very diverse functions. They can fight off bacterial infections, they can fight off viral infections, they even play a role in fighting off cancer in our bodies. So they have very diverse functions. As we look at those lymphocytes, they can destroy human cells. It's the only white blood cell that can specialize in neutralizing the cells of our bodies. Why would we want to destroy a cell of our body? Well, if we think of it, cancer is when our own cells are reproducing out of control. And in those situations, it's to our own benefit at the organism level to destroy those particular malfunctioning human cells so that we as an organism can have a better outcome of life. Or if we have a human cell that's infected with a virus, that human cell is toast at that point. And you can either kill the cell right away and minimize the number of virus babies or viron particles that are produced, or you can let it linger and stagnate and pump out lots of virus particles. Either way, the cell itself is toast and dead, but the difference is how many other viruses are you going to let it produce before it dies. So these lymphocytes will kill human cancer cells and human cells that are infected by viruses. They also are capable of presenting or showing antigens to other cells of the immune system to help activate them. And they help to coordinate actions of other immune cells. And then finally, lymphocytes can differentiate to make into other cell types that will make antibodies or give us the immune memory, so we can have that wonderful secondary immune response. Our other agranule site is the monocyte. Mono for one, it has one nucleus in it, and monocytes generally are going to increase in numbers when we have viral infections and or inflammation. Monocytes are known for leaving the bloodstream and then transforming into the big eater, the macrophage. Macro for big, phage for eat. And these macrophages will phagocytize pathogens or cellular debris that needs to be removed. They help to clean up the neighborhood. And the macrophages, after they clean up the neighborhood, after they phagocytize the bacteria and cellular debris, will take the bits and pieces and present them to other immune cells to help activate the immune response. As we look at the life history of a leukocyte, the process of making white blood cells is leukopoiesis. Erythropoiesis is making a red blood cell. Leukopoiesis is making white blood cells. So we're going to start off with the hemopoietic stem cells, the hemocytoblasts. And there are some different kinds of cells that those hemocytoblasts will differentiate to. They can differentiate into myelinoblast, monoblast, or lymphoblast. The myeloblasts will form the neutrophils, eosinophils, and basophils. The monoblasts, well, as the name implies, make monocytes. And lymphoblasts, as you guessed it, make lymphocytes. All of these cells are going to have reserves in the red bone marrow. And then those granule sites and monocytes that are stored within the red bone marrow are going to be released to the rest of the body. The lymphocytes, notably the T lymphocytes in particular, are going to be finished off or have development completed within the thymus. And when you think of T lymphocytes, I want you to look at that T and think T for thymus. The T lymphocytes are going to mature within our thymus. Now, as we're looking at these white blood cells, here's a flow chart showing the process of development for our white blood cells. So we start off with our hemocytoblast, our generic hemopoietic stem cell or multipotent stem cell, and then it can pre-differentiate into that lymphoblast, the monoblast, or the eosinophil blast, basophilic myelinoblast, or the neutrophilic myelinoblast. The idea is that we start off with one master cell that can then become any of the white blood cells. As we look at the white blood cells and how long they spend in the bloodstream, granule sites typically are going to leave in about 8 hours and then live 5 more days after exiting the bloodstream. And those granule sites are the ones with granules, the phils, the neutrophils, the basophils, and the eosinophils. Monocytes can leave the bloodstream in about 20 hours, turn into a macrophage, and then live on for a couple years. And then lymphocytes that give us long-term immunity can live for decades. And they can be continually recycled from blood to tissue to lymph back to the blood to help refresh that individual cell. If you have any questions or comments about the white blood cells, please feel free to post them in the class discussion board or to shoot me an email. And as always, happy studies.